got a few people online as well, and we'll put them up on screen at the end. Vic's going to talk for about 40 minutes, and there should be plenty of time for questions and discussion. Although I need to finish, I understand, at 5:02, because there's another microbiology session in 2 buildings down at 2 o'clock starting. Yes. So Mick needs no introduction, I think, to anyone in this room, but he's a senior lecturer here in the ESI, but also in the ECEHH, still can never get it right, in Truro. But Truro colleagues are going to be moving to our campus, hopefully, in the coming year, and we're based in Peter Lanyon, so that'll be scope for much more two-way traffic. And that stands for the European Centre for Environment and Human Health. Mick came to Exeter in 2012, so he's been here over 10 years now. And was just telling me that he started in the TIC building over there before ESI was even open. So he's a proper long service member of staff. He's had a very exciting life as well. He did his PhD at Eberhard Karls University, which I hadn't heard about till today. It's in a place called Tübingen.
Yeah, it was actually at the Max Planck Institute, but you have to have a university degree, of course. I wasn't actually based at the university. Yeah, that's old university. Yeah.
Yeah. That was in 2006. Then he had 2 postdocs, first one in Oxford with a certain Buckling sitting up there, I believe. Then he went to Wageningen. Yes, that was Wageningen. Then he came to us in 2012, so we're really blessed to have him. Since then, his career has developed leaps and bounds. He's a microbiologist focusing on evolutionary ecology and health implications, things like AMR. But interestingly, his first degree was in oceanography, which I hadn't realised. So he's moved from, well, oceanography, ocean biology, marine biology. So he's kind of moved a bit into microbiology. He's a key member of our microbiology group here in this building, of which we are very proud. And he, I think, does lots to connect up lots of the conversations that are happening across disciplines. So he's a real champion for what we're doing in the labs upstairs. That's enough for me. He's going to talk for 40 minutes on bacteria, sex and death. We're going to have a very exciting time with loads of great pictures because he's a brilliant photographer. And then we'll have Q&A. So thanks, Mick.
Thanks for the kind introduction, Jane. And I guess with my title, it can only disappoint from here, but I'll do my best. Yeah, it's really nice to kind of take stock over what I, did the last 10 years. And when kind of reminiscing, I went down memory lane and realized that actually I have come full circle because I was in Falmouth on the other side of the pond for my master's degree in the Wood School Oceanographic Institution. Very nice place, as you can see. Doing something completely different, looking at the deep sea taxonomy of deep sea mollusks. I have a photo, but a slight trigger warning. If you're offended by really obscene looking invertebrates, then you have to look away now. But yeah, so that's my, that was the species I was working on. I was actually unemployed for a year after that. I couldn't, strangely enough, find a job in deep sea taxonomy. I left that off my CV, but yeah, so that was the reality of it. I lived in landlocked places ever since. So when I came back to Cornwall, I was really happy to do marine biology kind of more as a hobby and see all these weird animals such as stock jellyfish and fish egg eating nudibranchs, and especially seaweeds, which I really like, and kind of do marine biology as a hobby. But I quickly kind of figured out that I could do a combination of microbiology and marine biology and find reasons to Skype, and when the weather was good to go to the rock pools in work time. So this is an undergraduate student, Abby Kolklove, who did a nice study where she collected 50 types of seaweed, different extracts, and coupled them to 20 or so different Staph aureus clones with different multi-drug resistances from the local hospital from actual infections, and paired them up and found that more resistant Staph aureus was also more resistant to these seaweed extracts, indicating kind of cross resistance, which I will talk about later. But also interestingly found some extracts were actually best at killing the most resistant strains. So that was kind of my first step back into marine biology. And I have together with Mahasweta Saha at the PML and also Chris here in the audience, Shona Kaur, who's a really good PhD student, who is based at the PML and looks at seaweed microbiomes. So that's a really interesting combination of microbiology and seaweeds. We also just were awarded a summer CDT student looking at nature-based solutions. So whether seagrasses and and macroalgae can have positive effect on water quality. And I'm more tangentially involved with microalgae and pathogens, another kind of PhD project with Anne-Marie, who is also based at the PML. So there's a kind of strong PML link, and I might be a marine biologist in a couple of years again. As Jane mentioned, I did my PhD in Germany with this kind of my favorite bacterium, Myxococcus. So this is a soil bacterium that lives in kind of swarms through soil and eats other bacteria. So it engulfs its prey and lyses it collectively. When food runs out, it builds these multicellular fruiting bodies that you can just about see with the naked eye, presumably for dispersal. And if conditions are positive again and there's food around, they start their multicellular life cycle again by swarming. And so this creation of fruiting bodies is a cooperative effort where most of the cells die and only a minority end up as kind of hardy spores within the fruiting bodies. And it's possible for cells to cheat and be a spore more often than you would expect and increase in the population that way. And so this was all known from a lab strain, and for my PhD, I was interested whether myxobacteria in soil in natural populations, as you can see here, if different swarms, how different they are on a scale where they can actually meet and interact, and if they meet, whether they also can build fruiting bodies together. So I found that whereas strains were more different the further away you went, even at this scale of centimeters, there were many genetically distinct Myxococcus strains, and they definitely did not want to cooperate. And since it's my favorite bacterium, it's also kind of the taxon of interest for the NERC grant that we're currently running, which uses sequencing instead of cultivation-based approaches. And here you see a tree that Dan made, where there's thousands of different myxobacterial taxa, and you can see what taxa they are on the outer ring and what habitat they were sequenced from in this inner ring. And you can use all kinds of fancy phylogenetic methods to detect bursts of diversification. And we're trying to link those bursts with habitat transitions and also with the switch between specialists and generalists. I won't say too much about this because this has really been driven by Dan, who now, of course, is an independent research fellow. And hopefully when it's his time for the state of the art, he can tell more about this project. Also, it's not finished. So what I want to talk about for the next 30 minutes are two things. So first, an old fascination of mine, sex in bacteria, which I first discovered when I was sequencing these closely related Myxococcus strains and found evidence of recombination in their DNA sequences. And since I'm at a medical school, I want to end with not death of bacteria, but death by bacteria. So opportunistic pathogens. So with sex in bacteria, you might think, especially when you're not a microbiologist, what's that about? Because bacteria, there's no male and female bacteria. They're very good at clonal reproduction, so they don't need sex to reproduce like humans do. They don't do meiosis with crossing over, and so the mechanisms are also very different. But if you look at sex in its most kind of basic definition or broad definition, it's the coming together of genes from different individuals into one other individual. And in that sense, bacteria do have sex and are very creative in their sexual lives. So they have sex within species, but also between different species. And there are very different ways in which they can exchange genetic material. And it's not technically exchange because it's unidirectional. So there's a donor cell here. There's some space in between cells, and this is a recipient cell. And there's a couple of main mechanisms. Transformation is one. So free DNA ends up in the environment as cells lie. So sometimes they actively secrete DNA, and the recipient cell can take that DNA up and incorporate it into their chromosome. There's also phages shuttling around, accidentally packaging host DNA. And another main mechanism is the transfer of plasmids, where a sex villus is formed, and these circular elements are transported as single-stranded DNA to another cell. And there's other mechanisms as well. So there's this vesiduction, where little kind of bleps, they're called membrane vesicles, but all emerge with another cell. And there's new mechanisms being discovered. There's also, it's not as clear cut. For instance, natural transformation is able to take up plasmids, and sometimes it's hard to see what's a plasmid and what's a phage. So it's usually complicated. And I want to talk you through 3 examples of bacterial sex, all done in collaboration with foreign collaborators. So this is with a group in Slovenia, and we got talking because I found that my Myxococcus were not cooperating together, and they formed these kind of incompatibility patterns where different swarms would not meet. And they had the same thing with Bacillus, which is a completely unrelated soil bacterium. And here you see 2 identical or near identical strains being inoculated, and they merge together. But this is a different strain, and you see this clear zone of inhibition with lots of dead cells in the boundary. And so there's many reasons why they could do this. They could be territorial, or they could try to prevent exploitative genotypes to enter the other swarm. But I reckoned that this could also be a site of sex, because it doesn't really make sense if you're in your clonal swarm to have sex because you would replace like with like. It also, if you encounter a different species, wouldn't make much sense to have sex because the genetic variation is more often than not incompatible. But it would be good to have sex with another strain that is genetically distinct, but also thriving in the same environment and potentially having useful ecological adaptations. So they did this experiment where they paired 2 different strains. So these are 2 non-kin strains, non-related strains where you see zone of inhibition. These are very closely related strains, and this is a pure control with exactly the same strain, just differing in the antibiotic marker, so spectinomycin or chloramphenicol. So each of the strains has a different marker. You can then swap the middle of the plate and plate out on agar with both markers to see if any recombination has taken place. And you can see that indeed it was the case. In the least closely related strains, there was significant recombination going on. And so this was the site of sex as predicted. And this is a nice illustration. So this is set up with a plate containing no antibiotics on one side and both antibiotics on the other side. And if you inoculate both strains and you let them swarm together, and in this zone, you get these recombinants with both antibiotics, they're able to swarm over and colonize this novel adaptive zone with 2 different antibiotics. But of course, this is just an example of one recombination event cherry-picked that actually has a positive effect on fitness. And it's not evidence for sex itself being beneficial, because of course you could imagine that there's also lots of events where you create a less adaptive genotype. So Macaulay Winter, who's sitting in the back here, PhD student, together with Angus and a group in Norway, did an experiment to look at whether sex could be beneficial. So he took another species and experimentally evolved them for a number of generations. And he took the wild type able to exchange DNA and a clonal mutant, and he evolved them in medium to which you can adapt, but also medium with other species, which are likely to exert an even stronger selection pressure and a varying selection pressure, which makes sex potentially more useful. So after this experiment, he competed the evolved strains from these 4 different combinations of treatments against the ancestor, where you see the fitness is relative to 0. And you can see that actually the sexual wild type did not adapt faster than the control or the asexual control. And also there was no effect of the biotic environment relative to the abiotic environment. So it's not that simple to, I still believe that there is a sex benefit of recombination in bacteria, but it was not apparent in this experiment. Just 2 slides on a second chapter of Macaulay, again on species interactions, but on the ecological rather than the evolutionary timescale. So what he did is he had Acinetobacter and mixed that with a DNA marker and then plated it out to count the total number of Acinetobacter and the number that had taken up this DNA marker to get the transformation frequency. But then its actual treatment was to also add another species to see if there was an effect of other species on this kind of sex phenotype in Acinetobacter. which is, of course, important because bacteria don't live in isolation, they live in hugely complex communities. And these data, we're going to maybe analyze it this week or next week. We'll have to figure it out, so I won't say too much about this, but these are all kinds of different soil bacteria isolated by ELSE that were paired with Acinetobacter, and you can see that there seems to be some significant effects decreasing kind of sexual behavior of Acinetobacter. And then this is my final example of sex. So this is not the most technical slide I have, so don't worry. So these are streptomycetes that are not growing as individual cells, but rather in these long hyphae separated by the septa. And in their chromosome, they have these AICs, these genetic elements that can insert themselves in the DNA and excise themselves from the DNA and replicate, and also code for a protein that forms a pore that connects these different cells and are able to move from cell to cell. So this is also a conjugation, but very different from the conjugation you might be used to, because it's not a sex pilus and it's double-stranded DNA that transfers. And so student Caroline Chouffat, who I co-supervise, did an experiment. What's actually really interesting, if you mix two different streptomycetes on a plate, wherever they do this behavior and these AICs transfer, you see these POCs. So you can see sex events with the naked eye because there's a growth retardation at the point of POC of transfer. So what you can do, as in the previous experiments, you can combine two strains. These are the two outer rings, each with a different marker, and then you can select for strains with both markers. So these are recombinants. And we can see that these AICs, that might not be very visible here, hop from strain to strain. But we also see, that was known, but for the first time we could see the extent to which these AICs also mobilize host chromosomal DNA. So they're able to co-transfer huge chunks of DNA. In one case, more than 35% of the chromosome was replaced in one step in this sexual event. So a huge effect. Sex in bacteria is not purely piecemeal, but can have large genomic consequences. And that leads me to another topic I'm interested in, is that of the pangenome. So when people started sequencing multiple strains within a species about 2 decades ago, they saw that if you look at the gene content of these different species, that only a part of the genes are shared by all species, but actually most genes are present in only one or a few, but not all strains. So this was dubbed the core genome that's shared by all strains, and the accessory genome is the part of the pangenome that is not shared by all strains. And if you sample more genomes, of course, the accessory genome gets bigger and bigger. In the case of E. coli, in the tens of thousands of genes. Also, the core genome gets smaller and smaller, so it's only in a matter of hundreds of genes. And individual E. coli have about 4,000 to 6,000 genes. So in the beginning, people were making lots of these graphs and characterizing whether pangenomes were open or closed, and coming up with very elaborate Venn diagrams that are not necessarily helpful, I think. But in more recent years, people have actually tried to understand the evolution of pangenomes rather than describing them and making Venn diagrams. And that's important because these gene content changes, because of horizontal gene transfer and also because of gene loss, are likely to have much greater effects on fitness than simple point mutation. So this is a distribution of fitness effects. So any change to the genome can either be bad for you or be good for you or not really matter much. And you can imagine, and if you take up an entirely new gene in one go, or even 1/3 of the chromosome, that it can be really good, but also really bad compared to point mutations. And this is an illustration, again, taking these mixed coccus clones that I isolated during my PhD that were very closely related, and that's important, and you'll see why. And having 22 of these genomes and taking 11 independent pairs and lining up the genomes, and then counting the number of changes in the core genome, amino acid changes plotted here, and counting the number of gene content differences in the accessory genome. So you can see if the accessory genome changes at the same rate as the core genome. You get this line here, but that's not what we find. So we find that the rate of change of the accessory genome is maybe 2 orders of magnitude higher than that of the core genome. So before any mutation pops up in the core genome, many accessory genes could have been gained or lost. So it's a very fast process. What you can also see is that this slope is quite low. So it means that there are on average fewer gene content changes between things that are less closely related and more that are very closely related. And this can be explained by the fact that if a lot of gene content changes are deleterious or not good for fitness, they will be removed. And so these pairs here are further apart evolutionary, so they had more time for these changes to be removed than the strains here. So there's different fitness effects, as you would expect, but we don't have a really good idea how they are distributed. So we see that lots of these accessory genes are very short-lived. In the previous graph, you can also see that they are being removed over time, a lot of them. But if you would ask a microbial ecologist and say, here are 2 different strains from 2 different environments and they have 2 different genes, they would be like, well, sure, they're doing different things and they need different genes. And there's, of course, something to be said about different adaptations in different niche specialists. So that must be true as well. But there's also bioinformatic evidence in a paper that Elsa and I co-wrote that point at neutral evolution. So it doesn't really matter much. And there's actually also evidence from experiments where people shoot in DNA, random DNA in a strain and measure fitness and see that there's actually not a huge effect. So how to kind of work out what the proportion of these different fitness effects is difficult, because with population genetics, it's based on lining genes up and scoring things like synonymous and non-synonymous mutations. It doesn't really, there's no framework for accessory genes. You can't readily compare them. Like in, I just kind of move that into showcase a paper where we actually developed a new population genetic test, but that was based on synonymous and non-synonymous polymorphism. So that's not possible for these kind of genomes. So with Adam Ayer Walker and others, a colleague in Sussex, we're developing a new approach to estimate what proportion of accessory genes belongs in each of these 3 corners of that paradox. And that is based on the frequency of genes and also an estimation of their age and whether they are more or less frequent than you would expect when it was just their age and neutral evolution. Okay, so that was the more esoteric part of my presentation. And I want to switch to pathogens, since I'm at a medical school after all. And it's actually interesting. I'm not doing it because I have it. And so we were talking about pathogens and studying pathogens. And one of the questions, of course, is, how many pathogens are there? Because if you want to study something, you have to have a good idea of what the diversity is. And what is a pathogen? Because there's also, some people say that pathogens don't exist because something that might cause disease in me might not cause disease in someone else. So we've been kind of, so it depends on your definition, depends on how hard you look as well, of course. I think we looked pretty hard, was together with an undergraduate student who did this over a summer, having Parv, who did an amazing job, and with Dan and Luke as well, and a collaborator, pensioned collaborator from Trelisk. Anyway, so we used a pragmatic definition. So if something was classed as a pathogen in the citing literature, we would call it a pathogen. Does anyone want to hazard a guess at how many bacterial pathogens there are of humans? I mean, it's published, something I've seen. So we found 1,513 species, which is an amazing number. It's about 7 or 8% of all described bacteria, but that doesn't really say as much about the prevalence of pathogens, but more that we have only described a small subset of bacteria and mainly the things that are relevant to our health. So Dan built another one of his fancy phylogenies, and you can see that these species are scattered across different phyla and different classes. What we also could see, that made this accumulation graph where you could see the first pathogen was described in the 1820s, Serratia, and then you see a rise when kind of microbiology develops as a science and people invent petri dishes and so on, and a more rapid rise when there's molecular methods. But you can see a really rapid increase in the number of known bacterial pathogens. And I think this is not because there's all kinds of emerging species, but just because we're much better at telling them apart. And one example is actually this species. So John Lee, or the staff whisperer, as I call him, he could see kind of colonies that were slightly different from Staph aureus, and we went on sequencing those, and we discovered it was a new species, which we called Staphylococcus corneliensis, so named after Cornwall. So that is one data point in our graph we contributed to ourselves. And this was, I guess, you know, the pangenome stuff is really kind of gives you a headache. It's really deep stuff. And this was a lot more simple because basically it was a really long list of species, and that's pretty much what it is. But it was good to see that microbiologists liked it. So within a couple of weeks, it was part of the official bacterial taxonomy database, and also people in Holland added it to their AMR package for R, and we're very pleased. That was done, I don't know, within a week or so after publication. And this list is nice, but it would be nice if we could keep this list updated. I don't want to put more students through updating this list. And I got talking with a colleague at Computer Sciences, Fabrizio Costa, and he was very enthusiastic, and he said it was very doable to use machine learning to automatically update this list and host it in Exeter. So we're going to write a seed corn grant, and we'll have kind of hopefully a global open access database for pathogens, and we can do all kinds of stuff with this. So probably there's lots of metadata, timelines. We can do geography, different pathotypes based on their origin, for instance. So that's quite exciting. And this is just a link because we like open science, and there's this event going on tomorrow soon. All right. So there's lots of different pathogens, but how important, how relevant is that? I guess in the developing world, it is really relevant because you've got things like TB and cholera and Shigella causing really significant mortality. Here it's less so. So there's a graph where you can see through things like sanitation and antibiotic use and vaccines that actually the burden is quite low. I don't think many of us are particularly worried about bacterial infections. However, you could make the case that bacterial infections could be more important in the future, maybe due to the global antibiotic crisis, which means that infections are harder to treat. And I won't say much about this because you've heard from Anne and Amy and others about this, but also threats such as climate change and other anthropogenic pollution. So I do all of my teaching in planetary health instead of microbiology, which at times is quite depressing. And I also always cover some microbiology. And there's a lot of examples in where both the prevalence and exposure risks to all kinds of different pathogens go up due to climate change. So you have things that are uncovered when the permafrost retreats, like anthrax. There's Burkholderia that really likes kind of wet and humid and hot climates, and that's increasing. It likes saline soils as well, so that's an increasing problem with sea level rise. You have things like Legionella causing Legionnaire's disease, likewise thriving in extreme weather. So after extreme rainfall, there's spikes in case of Legionella, probably because of aerosolization and people breathing in Legionella that's released from the soil. And also in the ocean, there's Vibriosis, Vibrio bacteria that cause very nasty infections and are associated with disasters, of which there will be more. So I guess it is crucial if we want to know about our exposure risks to environmental pathogens, that we need to know where they are in soil and freshwater marine environments and what species we can find in these environmental reservoirs. And I guess we've had two main approaches to quantify them: culture-based approaches, such as these plates, for instance, for E. coli. But that's quite different because there's 1,300 different, or what was it, 1,513 different pathogens. So you need lots of these expensive different agars to kind of cultivate all known pathogens. Or you could do sequencing, but that's also not perfect because you might see, species that are known to cause disease, but they might not be actual pathogens because their virulence genes might not be active, for instance. So it's not a direct measure of virulence. And we had a brain wave to actually isolate pathogenic bacteria based on a relevant phenotype, namely virulence. And that was very simply taking samples of water or soil washes and inject them in these wax moth Galleria larvae. And so the idea is very simple. If there's pathogens in this sample, then the larvae will die. And if there's no pathogen, they will not die. So it's not rocket science. It's basically injecting literally shit into maggots. That's what it is. But I was quite excited by it because it's quite useful. So I had a visiting student, Rafael Hernandez, with me for summer, and we set out to test this idea, and we took samples to different occasions, 4 beaches and 4 harbors around Falmouth, and we collected in each site water and sediment, and we injected those water samples in Galleria to see what would happen. And we also had control, so we filtered the water, so to control for any pesticides or any toxins that would go through the filter, and bacteria wouldn't go through. And so we took all these samples, sediment and water, injected Galleria, and monitored their survival for 3 days. And this is what happened. So you see the 2 different sampling dates, 2 different sampling types. And you can see in some cases, there was maybe 1 Galleria died, but everything seemed okay. And in some cases, lots of Galleria died. So there was something going on in those samples. And importantly, those mortality controls, those filtration controls, nothing died. So it must have been bacteria and not something else. So now we're interested. Okay, we know that some samples obviously contain pathogens and others don't, but what are these pathogens? So we took kind of the 4 samples with the highest mortality, and we plated out the hemocoel of the infected larvae on a plate to be able to pick the bacteria coming out of these infected poor wax moth larvae, and then re-inject them at 3 different CFUs, so 3 different densities, 10, 10,000 or a million cells. And then we genome sequence as you do. Okay, so this was the first species, Proteus murarius, and you can see that was really virulent. So after 15 hours, all the Galleria were dead, even when you would just use 100 cells. And we sequenced the genome, and we saw a Salmonella genomic island present in this Proteus. So this is actually a nice link to the sex part. They've taken up a bit of DNA, or a large bit of DNA actually, from a completely different species. To my knowledge, not encountered in the UK before, but known from hospital outbreaks in other places, in China and France. So the second species was really not as virulent. And this was a species I'd never heard of. And then I looked, and it was only found once before in 2 septicemia cases in South Korea. So people were very ill with this, but it was not found anywhere else before. So it's present in Korea and also on Gili Beach. And you can get septicemia from it. So that was nice. It's not something that we would have ever found by sequencing or just plating out Vibrio. And then again, a thing that was more virulent, kind of a usual suspect, E. coli, with more than 200 virulence genes. So all kinds of virulence genes being able to colonize different body sites and do damage. So that's a really nasty bug. And then finally, also kind of a usual suspect, Pseudomonas aeruginosa, I think it's ear infection or something, not as virulent, but with many dozens of different resistance genes, which are highly relevant ones, as I was told. So we followed up this approach with soil bacteria and also with marine plastics, and found more, recovered more pathogen diversity there. Okay, so this is the final 2 examples in my talk. And this is less about ecology, but more about evolution. So we know that our effects on the planet can have effects on bacterial evolution. And one example is that of co-selection for antimicrobial resistance. So you can imagine that if there's lots of metal pollution, bacteria evolve ways to deal with the metal pollution. For instance, efforts pump out metals. And these same adaptations can also serve to pump out antibiotics. So by polluting with metals, you can make things metal resistant, but also antibiotic resistant. And pollution is rife. This is a map of the northwest of the UK with all kinds of pollution events and industry. And we obtained a sediment core via our geography colleagues to look into more detail. So rather than comparing a polluted site with a non-polluted site, what we did is not take a spatial, but a temporal approach. So we looked inside the core and to document pollution through the ages and how bacteria were affected by that. So this is actually not that location where we got the core from, but this was just in Cornwall where we're trying this approach out with different student projects, a Macareth core, what it's called. So we obtained a core from Northwest England. So this is what it looks like. And my student, Andy Dickinson, and by rest student, sliced this core in 50 different slices. And what he did was a number of things. So he looked at all kinds of metal concentrations alongside the core. He did whole community sequencing through the core. He did qPCR of a marker for antibiotic resistance to the core. But what I think was most interesting was simply looking at culture-based approach. So he was isolating bacteria from these different slices. So reviving bacteria from a while back. So this is actually the only graphical abstract that has ever come in handy. This is the age of the core. So you can see it's quite almost 200 years of evolution that we covered. And this is the zinc level. So we took one of the metals, we quantified more, but zinc is one of the more toxic and common ones. And you can see it peaks around the 20s and 30s. Then what he did, he plated out bacteria on normal agar and agar amended with zinc. So he had a ratio of zinc resistance. And you can see there is a peak here, and he did the same with an antibiotic, cefotaxime, which is a clinically important antibiotic. And you can see that there's a nice correlation between resistance to zinc and cefotaxime. Moreover, cefotaxime was only introduced in the hospital around this time. So this cefotaxime resistance predated our use of that antibiotic. And then finally, so we know quite a bit about co-selection for AMR, but not really about co-selection for virulence. And of course, bacteria that are both virulent and resistant, that's where the problem lies. And we have plenty of metal pollution in Cornwall as well, of course, is the Carnon River. And so the idea was, in Luke's project, the bacteria living in these environments, such as Pseudomonas, produce these siderophores, and they're surrounded by toxic metals. And these siderophores are molecules that chelate these metals and prevent them from being taken up, and basically are detoxifying them. So that's an important function. But I guess the most well-known function for these siderophores is that they're usually quite specific to iron. And in contrast to these other metals, they're receptors where cells can actually bind these siderophores that have taken up iron to take up the iron into the cell, because iron is often limited for growth in bacteria, and especially when they're growing in a host, because we actively withhold iron in our body from pathogens to prevent their growth. So there's two roles of these siderophores. And our hypothesis was, if you select for siderophores for detoxification, they might be better at stealing iron from the host as well. So this is the experiment that looked at, so you took Pseudomonas aeruginosa, evolved it, not for a very long time, but enough to see significant changes without copper in this case, with low concentrations or in high concentrations. And after the experiment, he quantified siderophores and again used the Galeria system to measure how good they were killing their host. So first, both after 2 days of evolution and 12 days of evolution, you can see that siderophore production was increased as a function of copper concentration. So that fitted our prediction. And then he injected it into Galeria. And as you expect, there's also more mortality in Galeria in the treatments with higher copper sulfate mediated by this production of extracellular fluids. And I see I'm at 39 or 40. So this was the last case I wanted to present to you, which leaves me with my acknowledgement slide. I want to thank all the many people. It was actually quite nice to see how many people actually passed through the lab over the years, and also my collaborators in Exeter and elsewhere, and my lovely colleagues at the European Centre and the ESI. Thank you.
I think that was wonderful.
The timing was good.
The timing was perfect. Anybody got anything they'd like to raise? I keep thinking about those little creepy crawlies, but yeah.
Yeah, thanks, Nick. Really enjoyed all of it. I'm interested in this, what you call paleontological approach with the course. I'm not familiar with it at all. How do you know that? Obviously, you can look through the pollutant levels throughout the years. But how do you know that the bacteria found in these different segments of the course aren't all modern day bacteria that just migrate through and just adapt to the levels of pollutants that are already present other than historical record?
Yeah, I guess we can't completely exclude that. I mean, that core was very stratified, but I don't know if that probably doesn't mean that bacteria can't go through them. That is true. It would still show co-selection.
Yeah.
I guess so. In that sense, it's not a problem. But yeah, I see your point. That's a good point.
Yeah, no, I was just curious.
Yeah.
Anything else you do to like revive?
Yeah, and we didn't actually even know. So that was my main worry with that paper, because we didn't do 16S on the isolates. I don't know if they're spore forms or not. And I was very, and we didn't have the isolates anymore. And I was very worried that the reviewer would say, "Well, it's speak," but they ignored that. So I was really happy.
I had no idea you were injecting caterpillars or bugs up there.
Yeah.
That was a big eye opener.
I'm not the only one in my defense.
Yeah.
So
And is that a standard? Is it a standard?
well, so it is. Yeah, there's thousands of studies. I guess it is standard to take a clone, so to take different strains and compare their virulence, see whether certain cholera strains or whatever are more virulent, or to have a knockout strain. So if you suspect that a gene is involved in virulence, you can knock it out, and then you can test whether it actually has decreased virulence. So the novelty of this approach was to not inject with a single strain, just throw the whole microbial community in there and let the pathogens kind of select. So we incubate these at 37 degrees, and insects have a very similar innate immune system to humans. So generally, in a few studies, it correlates. If things are more virulent in the Galeria system, they're also more virulent in a mouse system. So you don't have to kill mice, which is good.
Yeah, and that was great to talk. I really enjoyed that. I really liked a bit about the number of pathogens. So you said there were about 1,500 pathogens that you kind of looked at, and that's it's a really high number, isn't it? But then you also said that was about 7-8% of the total like known bacteria. But is that like what is our estimation of the total number of bacteria? Because that number seems quite high. And also we've looked really hard to find the pathogens, but have we looked as hard to find just general bacteria?
Yeah, I mean that's the whole kind of topic between the old school kind of taxonomists, and they have their list of whatever 25,000 species, and they're all culturable. So it can't be a new species if it's not cultured. And then you got all the kind of high throughput sequencing people that are increasingly able to reconstruct entire genomes just from the metagenome, and they see tremendous diversity, but they are not labeled with a species name because the taxonomists don't like it. So there's kind of tension there. But I guess there will be many millions of species, but kind of the estimates of bacterial diversity are many orders of magnitude apart. We don't have a good idea. But yeah, this is heavily biased, of course, to bacteria that are living on us or near us and causing disease. But still, yeah, it's a high number.
Yes, no, I agree. I think that's
And we'll find
Yes,
out.
websites seem like kind of like less than 1% of bacteria actually causing disease. But I think it depends what you call a species. because
Exactly,
what is a
yeah.
species?
It will be
Another
much
thing-
less than 1% if you take it to the true diversity account.
Yeah, let's
It's only one, yeah.
Angus.
Thanks, Nick. That's great. I was really curious, actually, just following on from what Andy's just asking, what percentage of pathogens are, I guess, real pathogens, obligate pathogens, that kind of do, opportunistic, and then what that then means for, I guess, the emergence of new pathogens with environmental change.
Yeah, I think that's really, I mean, I've been debating with Richard Bendel to follow this up and try to kind of make sense of what a pathogen is. And this kind of, people use opportunistic and obligate, but that's also not entirely right. So even obligate, tuberculosis, it's more likely to make you ill if you already are poorly. So they have clear opportunistic tendencies, even if they're classified as an obligate pathogen. So it's kind of a gray area. But I think maybe a better one is, the ability to transmit would be more of a feature of a specialized pathogen. And that's definitely a small minority in our list. I have evolved ways to transmit from host to host. There's even like chemolitho autotrophs, that can metabolize gold, that have caused infections in humans. So quite obscure. I mean, that's obviously not really a pathogen because it normally doesn't even eat organic material, but it can still cause disease. So we just kind of had, it was reported from the site of an infection, and considered by the authors to be a pathogen, which is not a definition that maybe an evolutionary biologist necessarily would use.
Maybe it's one of those things that highlights how much we don't know. The more you because you assume that people know
It's
these
almost-
things,
yeah.
but they
Yeah, what a pathogen is, it's not
Yeah.
a simple, it's not a simple question at all. No.
Mm.
And so I've been discussing with my collaborator to see if there are certain kind of key characteristics that are actually scoreable. So for instance, where you can transmit
Yeah.
from host to host or not, whether you can infect non-immunocompromised people or not, whether you can colonize without needing injury or kind of a medical device, but if you're able to colonize. And actually, after talking with Fabrizio about the maximum, the machine learning stuff, that might actually be something that we could get from the literature as well, automatically, kind of these measures of, and kind of make a PCA plot where different pathogens fall and where probably things that are completely unrelated might have a similar lifestyle. Maybe there's certain types that emerge, but that's for the future.
Actually, I'm reading if we should put up the online people and see if anyone out there. I forget they're there. Yeah, if anyone online would like to ask a question, we can now get you on screen to ask it or make a comment. And I was wondering what you were talking about sex at the beginning, because obviously these organisms transfer their genes so efficiently and is on such diversity. So even in evolutionary terms, they're quite advantageously placed for environmental change, aren't they? Because they can capture such diversity in such a short space of time. They've got so many means of changing themselves.
Yeah.
Human beings are much more complicated because it all takes place through biological sex, two sexes.
Yeah.
Changing genes is very slow.
Yeah, absolutely. Yeah.
And yet we're seen as the more advanced organism. So what do you have any feeling about that? I mean, why?
No, I don't want to wade into that. But I mean, the debate. No, I think bacteria are far superior to humans. Let's put it that way.
Because if environmental change comes very rapidly, humans will be wiped out, but bacteria are very well placed.
They'll always be there. Yeah,
Yeah.
absolutely. Yeah.
You could almost never get rid of them.
No, it would be impossible to sterilize the plant without blowing it up.
Yeah, it's quite an interesting thing.
All right. Thank you.
That was a great state of the art. So thank you very much. Thanks all for coming. Next month, it's the end of April, our state of the art, and Karen Scott's going to talk about homelessness and people living in vans. So a completely different topic, but also extremely interesting to make us think about the world we live in. So hope you'll all come to that. And big thanks to Mick. And if anyone wants to volunteer, I think there is a slot or two later in the year. All right. Thank you.
Yes.